Can you all see my screen and the laser pointer?
Yes, we can.
Yes, yeah.
Yes.
All right, perfect. So welcome everyone, and those of you that join us via the YouTube channel. Like I said earlier when we did our kind of round call, I'm Johnathan Mayfield. I'm a senior grad student in the Lance Wells Lab, and presenting with me today is Naomi Hitefield. So today what we're going to talk about is what the lab is doing, what we plan to do, and how that is going to hopefully better our understanding of those affected by mutations in OGT. See if I can get my slides to go. There we go. So kind of outline for this presentation, we're going to start with who's doing the work in the lab. So that includes Naomi and myself, as well as former members and other members in the lab. We'll do some brief background and basics of OGT, as well as OGT CDG, or also known as OGT XLID. I'm also going to go over what it is that the lab does when a new variant is reported to us, and kind of the information we get from clinicians and that we can provide to them. As of now, we don't have a diagnostic. Then we're going to go over what's been determined to be different between the variants and the wild type. And for this, we're going to stick mostly at the level of the actual OGT protein. And then we're going to look at what's next for our lab, with the goal in the future of getting to diagnostics and potential therapeutics. There we go. So the work in the lab really started with Krithika and then Nithya. So these are both former members of the lab. They characterized the first 5 variants that you see here. And all of those are in the TPR domain. And I'll get into what that is on the next slide. From there, Hannah in the lab, a graduate student, worked on characterizing what proteins were interacting with the TPR domain of OGT. And that work has been continued by Naomi, specifically looking at some of these variants, these mutations in OGT to see what is changing in terms of what proteins are interacting. The work that I've been doing since I've joined is characterizing these 3 new variants or mutations of OGT. Those are all located in the catalytic domain. And again, I'll talk about what those 2 domains are on the next slide. Laura, who, pictured here, she just graduated, but she's been doing work on these 2 here. And I'll note that this one here is the one, the Skripkin family is the one that reported us this variant. We've also got some new undergrads in the lab that I've been training. We've got Genevieve, Lila, another Lila, and Jordan. And so they're also working on some uncharacterized variants. We've got Jeff in the lab who is working on generating engineered cell lines that contain these mutations so that we can have a good model for what we're studying. And Alyssa in the lab is kind of working on the flip side, looking at OGA, and I'll get into what that does as well. There we go. So the OGT gene is one of the most conserved genes in the human genome. So that means between me and everyone on this call and everyone that's watching, for the most part, our OGTs are going to be identical. And then in terms of looking at humans versus other animals, whether that's mice, rats, even down to flies and worms, it's going to be very conserved as well. So across evolutionary history, this enzyme has been very important, and there's not been a lot of room for changes in the sequence of that enzyme, that protein. And part of that is that we know in mammals, at least, a loss of activity is going to be lethal at the embryo stage. So that means for everyone alive today that they've got a copy of OGT that works at least some. And before I get into kind of the structure of OGT, the kind of two big parts that we talk about, I want to note that you need the functions of both of those parts, the catalytic and the non-catalytic, for mammalian cells. So from left to right here, we've got this TPR domain. And so this is going to be for your interactions with other proteins. And it's also going to be for selecting which substrate or other proteins it's going to modify. And then on the right side here is the catalytic domain. And so that's going to be responsible for, especially in mammals, two functions. The first is adding O-GlcNAc, which you'll see as this blue square here, onto proteins. And I mentioned Alyssa earlier works on OGA, and that's going to take off the blue square, that O-GlcNAc. And so this is kind of the main function of OGT, is cycling on O-GlcNAc onto proteins with OGA taking it off. For mammals, OGT has also got another function that you don't see in species like flies. And that's going to be cutting this protein HCFC1 in half, which is required for the function of that protein. So next, let's see if I can get there we go. Kind of want to talk briefly about the clinical features that we see for OGT, CDG, or XLID. So broadly, it's estimated that 1% of the world population is affected by an intellectual disability. Within that 1%, you have those that have mutation in OGT that causes an intellectual disability. And we kind of gone back and forth between two names, either OGT, congenital disorder of glycosylation, which kind of focuses on the idea that there's an issue with adding that sugar onto other proteins. We're also calling it OGT XLID, so that we can kind of be grouped with other mutations on the X chromosome that cause intellectual disability. So with the OGT gene being on the X chromosome, it's an X-linked inheritance. So for typically females are going to be born with 2 X chromosomes, while X chromosomes, while males have 1. We have seen for some variants that you won't see an affected mother, but you will see her male children that will be affected by the ID. But then on the other hand, we have some variants, some patients that are female. So there's some more work that needs to go into understanding kind of what this inheritance pattern looks like. And one good thing that would help with that is understanding what is the male to female ratio. And that'll get into the kind of this big point in a moment. But in patients, typically what you're going to see is that they're going to have the intellectual disability, which is typically, you're going to say, an IQ below 70 and or limitations in adaptive functioning. So kind of doing those day-to-day activities, and then also developmental delays. There are also some other features that can show up but don't show up in every patient. So dysmorphic features, so like a full philtrum, which is this part of the mouth here, eye abnormalities, or some patients have fifth finger clinodactyly, which if you look at my pinkies, you can see that they curve away from each other. That's what that fifth finger clinodactyly is. So other kind of considerations that we're thinking about when we're studying this is that, like I said, we have male hemizygous, so that means of their 1X copy of the chromosome, they have a mutation on that one, or we have female heterozygous, so that means that they have 1 mutated copy and 1 normal copy of OGT. We have variants in both domains of OGT, so it's not localized to one area, it's kind of spread across the entire protein. And also, interestingly, we have variants that don't change the actual sequence of the protein. Rather, what happens is the regulation of that gene expression is altered by some mutations outside of what codes for the actual protein. And so what's really going to help all of this come together and give us a better understanding is a natural history study. So asking the right questions and asking them for a large group of people that are affected. So that's something that, especially we would want to work with clinicians for that. Because I may say this again, but I'm not a clinician, I'm not a doctor, I can't give medical advice. But the clinician would be able to know what questions to ask. And so what I've shown here, and you don't need to kind of memorize all of these, but these are the variants that either have been reported to our lab by clinicians or Dan reported based on his SFG presentation. So we're looking at around 62 known mutations in OGT that are possibly linked to intellectual disability. And so here in this first row here, these are, I've got initials for, like me, I'm working on this variant. Naomi's initials are here because she's working on these. We've got UG for the undergrads, so they're definitely helping to start characterizing more of these mutations. And then I've also highlighted some that Dan has published and working on. And then one other note is, and kind of goes to the back and forth that we have with clinicians, is this variant here, R627C. So when this clinician reached out, we asked, are there any individuals in the family, like uncle, father, that have this mutation and don't have that intellectual disability? And what we found for this one is there was an uncle that had this mutation but didn't have intellectual disability. And so what that tells us, if both of them have the mutation, but they don't both have intellectual disability, it suggests that maybe this variant, this mutation, is not what is causing the intellectual disability. But again, without a really good diagnostic, we just don't know for sure. So now we're getting into kind of what do we do with new variants, and then we'll get into what's different about the variants. Right, so without a diagnostic, we have to use different pieces of evidence to suggest whether or not it is what's causing the intellectual disability. So one of the first things that we can do is use softwares that will look at protein structure, the sequence, the conservation of the protein, and make predictions about whether or not those variants might be causal, right? So these are predictions, not a diagnostic. So what I've got shown here are two different programs, and based on the 62 or 60 mutations on the previous one that do change the protein sequence, they've been categorized. So red, it predicted that these are likely causing the intellectual disability. The gray is uncertain, so not many there, and then a few that are benign. And again, these are predictions. I know between these two programs, we had at least 15 of the mutations change categories. We can also check the conservation among animal species. So to orient you to this here, this is the letters here represent the different amino acids that make up this part of OGT. And I've got humans here. We've got chimpanzees, rat, mice, fish, flies, and worms, right? So a wide variety of animals. And what I want you to look for is black means that it's the same between human and that species. And as you can probably tell, there aren't a lot of changes, just a few, and they tend to be in the fly or in the fish here, right? So this tells us that, in particular, where this mutation has gone from a T to an A, that residue has been very important for a very long time. And that suggests to us that potentially this is pathogenic, that it's causing the intellectual disability. We also can check for unaffected individuals. So there's a a large database where they sequenced more than 10,000 genomes. And if we find a particular variant is also in that population in a person without intellectual disability, similar to the variant on the last slide that I mentioned, we might that might suggest to us that it's not causal because we know somebody has it that isn't affected. And then we can also model that variant, and that kind of gives us an idea of what we need to test in order to understand that variant better. All right, so now I want to get into what is different between our wild type enzyme, our protein, and the variant protein. And so kind of the big categories that I have here are stability and expression. So how stable is that protein? Does it unfold quickly? And then in that same vein, expression. Do we see a lower level of that protein compared to our wild type? Then that protein I mentioned earlier, HCFC1. So looking at the activity towards that protein. We also have OGA levels. So kind of that balance between OGT and OGA, looking at that. So glycosylation is another category. And so this is going to be the ability for the OGT to add O-GlcNAc onto other proteins. And then from there, Naomi will take over and talk about the proteins that interact with OGT, as well as what we've seen in terms of changes in what genes are being expressed. So to start, I just want you to kind of look broadly here, right? These don't look that different, right? When we run statistics, these aren't different. So this variant doesn't seem to have an issue with the same levels of protein as wild type. But when you come over here for this variant, you can see quite a large difference, right? And this is when we express this protein, it just never reaches the same level as our wild type enzyme. So that might be one area that is impacted, but it's not a common difference across the board, unfortunately. So just want you to look real quick at these numbers here, right? Wild type is 55, and we've got a variety, some of them really close to wild type. So even among this group here, we don't see a common difference. Then when we get into the activity of the OGT wild type and the variants towards this substrate, this protein HCFC1, what you want to look for is you've got two bands, so it's cleaved in half. We see wild type, we've got two bands. This particular variant, we see two bands. This looks very similar to wild type. However, when we look at this other variant, R284P, we see that we get a smaller band down here. Now, some other variants that Dan has characterized also see something similar, but again, it's not a common issue across the board. It is an issue for some variants, for some mutations in the protein, but what we really need, and I've mentioned this a couple times, is a way for us across the board to determine, are these variants causal? Are they causing the intellectual disability? Early on, with the first characterization that Krithika did in the lab, we thought OGA levels might be that marker that we can look for. So what we've got here is this is a control. This is within the same family, all three of these individuals. We have two patients that have a variant in OGT. And you just want to look for the black here, right? So that is the OGA levels. You just don't see as much for L254F, these affected individuals. So with that first one, we thought, okay, maybe it'll work. We took that same variant along with some others, and we engineered those into embryonic stem cells, so very early in terms of the development. And we didn't see a change there. And then there's also evidence suggesting for other variants that we don't really see a big change in the OGA levels. So unfortunately, again, not a common difference for all the variants. And what we really need is something that we can look across the board for. Then when we look at glycosylation, we do see kind of two categories, everything kind of going into one of two buckets. So this variant Laura was working on is in the TPR domain, so that left side of that slide earlier. We don't see a difference between these. The averages are quite close to each other. But when we look at variants of the catalytic domain, like the three that I'm working to characterize, we see a reduction in terms of the number of proteins that are being modified with O-GlcNAc. So we kind of have two buckets, the TPR domain variants that don't typically have altered global changes in what proteins are being modified with O-GlcNAc. Whereas when we look at catalytic domain variants, whether it's ours or the ones that Dan's group has worked on, we do see these global glycosylation defects. So I will note, especially when they're engineered into cells, a reduction in this OGA enzyme that takes it off can help bring that back to balance. And now I will turn it over to Naomi.
Yeah, so like Johnathan mentioned, OGT has, these two different domains, and he talked, looking at some of the ability of this protein to be able to add this O-GlcNAc with its catalytic domain, but I want to focus a little bit now looking at the TPR domain. And the TPR domain is important in these protein-protein interactions, really helping the enzyme select which protein is actually going to be modified with O-GlcNAc. And so one thing that I'm working on in our lab is investigating how changes in our mutations or OGT-XLID variants in the TPR domain actually can affect these protein interactions of OGT. And so we do this by using a dual approach to isolate proteins that are interacting with OGT. And then we identify these proteins with mass spec. And then we can compare which proteins interact with a variant OGT compared to which proteins interact with wild type and see how these interactions change. So far, I've looked at 5 different variants within the TPR domain, and we have some evidence that looks like these variants do indeed have altered protein-protein interactions. For example, we found that these variants have a decreased interaction with a protein Johnathan was mentioning earlier, HCFC1. And this protein is important in transcriptional regulation. So this is going to be something interesting to look into. And this work has a twofold impact. First, it gives us insight into the biology of how these variants could be causing this OGT-XLID phenotype that we see in our patients. But it also can provide potential biomarkers or targets for therapeutics. So in related work, we've also begun to study how gene expression can change, or if it changes when a variant OGT enzyme is present. And so far, we've looked at multiple variants in the TPR domain, and we found common up and down regulated genes changing compared to wild type. And these shared altered genes could theoretically serve as potential biomarkers for OGT-XLID. And this is a work in progress because we haven't yet tested this thoroughly for the catalytic domain variants. But this could provide us with a common link between these two subsets of patients, what Johnathan was talking about, the catalytic domain, patients with variants in the catalytic domain compared to patients with variants in the TPR domain. Or it could provide us with domain-specific biomarkers. So it's definitely a work in progress there. So before we move into what we plan to do in the lab, I want to summarize what we know so far in terms of what changes we're seeing with our OGT-XLID variants. So first, some variants have altered stability or expression, like we saw with the Y835C. Some variants have altered glycosylation and altered ability to cleave the HCF1 protein. Some variants have altered OGA levels in some cell types. In terms of glycosylation, the catalytic domain variants typically have altered levels of global O-GlcNAc, but this is not the same for the TPR domain variants. But for the TPR domain variants, we've seen that they have altered protein-protein interactions. And lastly, we've also seen that the TPR domain variants have common changes in gene expression, both in terms of genes that are upregulated or downregulated compared to wild type. But we haven't studied this for the catalytic domain variants yet. But overall, we're looking for a common change of biomarker in all variants, if possible, so we can start looking for ways to help all of the OGT XLID patients. So we characterize our next steps into two different categories. First, we're going to continue characterizing the OGT XLID variants, both at the biochemical level and in cell and animal models. And this work is important because it helps us understand the biology behind what's wrong. But secondly, we're taking steps to understand the basic science behind potential therapeutics. And we've only begun to lay the foundation for this, but it's important that we have good model systems, cell and animal models, to best evaluate potential therapeutics. So for characterization, we look at changes in the stability in the activity of the enzyme. And this is work that Johnathan and some of our undergrads are doing. But we're also generating lines of male neuroblastoma cells with a variety of OGT XLID mutations. And with these cell lines, we can look at things like changes in gene expression, which I was mentioning before, proteins, OGT protein interactions, and O-GlcNAc. And this approach will hopefully identify a common biomarker or diagnostic, either, like I was saying, domain specific or for all OGT XLID patients. And this is work that myself and Jeff in our lab are currently working on. And as we've said, this is important because a biomarker or a diagnostic test greatly improves our ability to both diagnose patients with OGT XLID, but also look into potential therapeutics. So one such therapeutic that we are just beginning to test is substrate supplementation with glucosamine. So the premise behind this is that essentially we hope that if we can raise the levels of UDP-GlcNAc, which is the sugar donor for OGT, we can actually increase global O-GlcNAc levels back to wild type levels. We've tested several variants, and we've seen that we can increase the O-GlcNAc levels of these variants closer to wild type. But we want to point out that there are definitely some caveats. First off, this may not work for all variants that don't have, particularly variants that don't have a global defect in O-GlcNAc, like our TPR domain variants. It may not reach the levels necessary, but the thing we want to point out is this work has been done in cells in a culture dish. And so we don't know if this will work when we get to animal models. So there are a lot of questions that we need to answer before this is a treatment option, but we're taking the first steps toward testing this avenue of treatment. One other potential therapeutic avenue we're investigating is the use of OGA inhibitors to help alleviate the global O-GlcNAc defect. So if you remember, Johnathan earlier was talking about OGT is the enzyme that adds the O-GlcNAc. OGA is the enzyme that can move it. And so like the premise with glucosamine supplementation, if we can add OGA inhibitors, we should be able to increase the global O-GlcNAc levels. But these have the same caveats as we discussed before. So these studies are also in the early stages. But thankfully, these inhibitors that we're testing have already gone through phase 1 clinical trials as potential Alzheimer's disease treatments. So that could be helpful in the future if these had worked in animal models and such. So the identification of an effective biomarker is imperative toward furthering our understanding of OGT-XLID. Once we have an effective biomarker, we can use different screens to test potential therapeutics or targets for therapeutics. So with a biomarker in a model that recapitulates the OGT-XLID phenotype that we see in patients, we can use things like genetic screens or a thing like an FDA-approved drug screen to identify targets for potential therapeutics, because by having our biomarker, we have a readout or something we can look at to see if it improves when we look at these different genetic screens or drug screens. So our key goal with everything that we're doing in our lab is to understand the biology behind OGT-XLID, to look for common changes amongst all the different OGT-XLID variants that we have, because we want to identify a biomarker to ultimately look into therapeutics that can help our OGT-XLID patients. So we want to thank each of you for the opportunity to come and speak about our work on OGT-XLID and what we're doing at the bench. And we hope that you guys were able to take away a sense of our path in the lab to better understanding this disorder, to help better diagnose and treat the OGT-XLID patients. And we are happy to take any questions that you all may have. And we want to thank Andre and Irina for bringing their family to the lab for a visit the other week. Thank you.
Thank you, Naomi. Thanks, Johnathan. This has been incredibly useful for us. Again, it hit me last time when you showed it. Now it hits again, that's good to understand that basically there is not one underlying change that we are trying to fix here, but multiple different options, and all of those options depend on a different variant, and the variant being in different domain there. So I have a couple of questions here. Can you, Johnathan, can you scroll back to slide number 9, please? So very basic question. The expression levels, expression levels. Just apologies for basic question. Expression levels, expression of what? Is it of what protein? I think you mentioned of a protein. It's not expression of glucosylation. That's not the one that was number like 4 and 5 or 5 in the list.
Yeah, so this is expression of the protein. So what we've done is created plasmids that we can put into cells that will tell them, hey, express this protein. In particular, we can look at the difference between when we do it with wild type and a variant. So between these two, and especially if you look at the actual raw data, they don't look different. But with this variant here, when we try to express it, even at 4 days of just telling the cells, hey, make as much as you can, that protein just will not express to the same level. We're talking maybe 25% of what we can get with a wild type enzyme.
Got it. So does it like does it mean that expression of the protein means how much gene produces this protein? Like the volume of protein that's been produced? Is it what?
Yeah, so at that particular time point, like a snapshot of how much protein is there that we can get. Yeah.
How is it different from glucosylation?
Glucosylation is adding
So
sugar.
this is the amount of protein, amount of O-glycanac transferase in the cell. And then you would think that will most likely correlate with activity. Not always, but oftentimes. It depends, right? So there are two different things. So you could have a normal amount of protein, but it could be like T578 is a good example. T578, the amount of protein is fine, but O-glycanac levels are down because it's not as active of a protein. It makes the protein, but the protein doesn't function completely normal. Whereas the Y835C is very, it's just not very stable. It doesn't express well, but what expresses is active. So did that make sense?
Yeah, yeah, it does. Awesome. I think the next one was now Naomi's slide, and or can you scroll next to the next, next slide? Yeah, cleavage. I think it was about cleavage. You mentioned that you observe the cleavage, and but there was a sentence that you've said that we need still to test it if it is causal to the intellectual disability. I'm not sure if it was about this slide or previous slide, guys. So I'm trying to understand here. All of those variants, they are causing intellectual disability, but you observe this particular change, and you need to prove that this particular change is causal to intellectual disability and not something else, right? Is that what it is? Is that the right statement?
Was it maybe one of these slides?
And it was later in the deck.
Because I know here I mentioned that these are predictions, right? So this doesn't,
Mhm.
nothing on this slide or that we do in terms of this can tell us for sure, like, yes, we know 100% that is what's causing it.
Mm.
Whereas these other kind of categories here, they provide evidence that, yes, they are causal. So, for example, on that slide, right? Like, this is not normal activity for an OGT. So this mutation is causing a defect here,
Mhm.
whereas you might see a different change here, and then another different change here, right? And it may not be the same across the board for every mutation from patient to patient.
Yeah, that makes sense.
And there was another slide with this intersections.
Okay. So, Johnathan, then my next question is about, do you know all of those parameters for all of the variants? So the question is, if you're saying that it's a glycosylation or cleavage or OGA, does everything else remain the same or normal as like wild type normal, or there are also abnormalities there?
Yeah, so I mean, it's just going to change depending on variant or mutation to mutation, and then also how you test it. So, for example, the OGA levels, when that was tested for the first variant that our lab characterized, they looked at patient lymphoblasts, so cells from the patient, and they found that OGA levels were down. But then we took that same mutation and we put it into a embryonic stem cell so that we have one genetic background. We don't have anything else that might be interfering. Compared that to wild type, we saw no difference. Whereas, this glycosylation, that typically is going to be your catalytic domains, and you're not really going to see that issue, especially globally, for TPR domain variants. One of the kind of most outside of glycosylation, the other area that kind of seems to point to a common difference is going to be this gene expression, right? We found, I think it was over 300 genes that were altered that were the same between 4 different variants. And that kind of points to that maybe one of those genes or a set of those genes might be common for other variants in terms of being altered.
I'm not sure I understand it, but so the mutation in OGT gene alters some other genes?
Yes. So OGT, in modifying other proteins, some of those proteins are responsible for what genes get expressed. So, for example, this HCFC1, its job is to regulate and tell the cell which genes need to be expressed when.
Oh,
And the amount of O-GlcNAc that's on there can change, depending on whether it's a mutation or whether or not you have, here we go, right? So it can be a difference in the O-GlcNAc on here, and that can change what genes get expressed. Or it could be interactions, right? Naomi's got a change in interactions. So OGT itself isn't like changing the DNA or causing like directly a change, but it's all these other proteins that it interacts with and that it modifies that do. So it's kind of a like big level or higher level regulator of gene expression by modifying these other proteins.
Got it. Now makes sense. Yeah. And there was a slide that shows intersection, talking about biomarker, and it shows intersection, some numbers. Can you explain us what we see on that slide? Yeah, this one. It points to this, areas with numbers. What does it mean?
Yeah. So the genes that are in the red circles are genes that are altered, either upregulated the 59 or downregulated the 379 in all 4 of those variants. So it's basically a giant Venn diagram that is looking for commonalities between the genes that are altered in their expression levels between all of the 4 variants that we have here. So like, for instance, that the 692 for L254F and the upregulated, those 692 genes are upregulated in L254F, but not in any of the other 3 variants. But like the 27 genes here, these are upregulated in both A259T and R284P. So there's commonalities between pairs of variants, trios of variants, and then all 4 variants. So we're looking at what commonalities there are between all of the variants.
So those numbers, yeah, that totally makes sense. So those numbers are gene numbers.
Yeah, number of genes, yeah.
Okay. And you're going to just go through all of those genes and just see how they react to like therapeutic or so to like substrate therapy or something.
So that's where this particular set of work here that we plan to do, right? So that was variants within the TPR domain. So we kind of have half of the protein. What we want is that for the entire protein, we have targets, right? So we're making, I think it's 13 different variants into the cell line. They include about half catalytic and half TPR. So we've kind of got a spread of mutations. And then we're going to repeat those experiments looking for a common set of genes.
But we're also, within those common sets of genes, we're looking for things like if they share, it gets more complicated, but if they share things like the same promoter, or if different transcription factors activate those certain genes, where they, are located on different histones. And it gets more complicated, but we're looking for like any commonality between like maybe how all of those different genes even are regulated or something like that.
Yeah, it might be, for example, like this group of proteins here might end up regulating, those 59 genes, right? And so this would be our target because we know that all of those genes that changed are because of that group of proteins there.
Oh, wow.
Thanks, Jonathan. I also see there's a question from Hughie. Hi, Hughie. You can actually unmute yourself and ask this question. There's a question in chat, and Hughie is from Dan's lab. So Hughie, do you have an opportunity to unmute yourself and ask this question?
Yes, thank you. Thank you for the fantastic talk. I actually have one question about on slide 9, also with the expression. Do you, when you measure the OGT levels, do you measure both the endogenous OGT and the transfected OGT, or you just measure the transfected OGT protein level?
So we have a tag on there, so it's just going to be the transfected. So it's not going to include the endogenous levels.
Do you think they have less expressions because for example, the Y2835 is unstable, so it doesn't express or be degraded faster than the wild type?
Yeah, that is a great question. So I have actually set up an experiment where I'll be in lab all day Saturday looking to see whether or not it is protein turnover. So we're going to use cyclohexamide, which is going to stop the cells from making any new proteins. and we're going to measure time points and see whether or not the this particular Y835C is just being degraded faster, and so it's unstable. So yeah, that is definitely next on my list of things to do.
Yeah, thank you very much. That's really fantastic. Really appreciate for your sharing. Yeah, thank you.
Thank you.
Yeah, and Huge, I know that Dan was presenting last time, but if you or your team members also have something to share, would be happy to create this space for you, because again, this is also an opportunity to collaborate between the labs as well.
Yeah, I think it's really good. We can share and also see what is going on, and to see I think it's a really good opportunity. Yeah, we would love to do that, I think. Yeah.
Let's absolutely do this. Neil, Sarah, Sage, do you have any questions?
No, I'm all right. Sorry, you guys.
Just one stupid question, but the WT term you're using, is that
Well,
sort of big? Is that base case is sort of how? I mean,
Yeah.
is if you described it another way, the WT would be base case, sort of the unaffected.
Yeah, so this is going to be our wild type. So this is, if we were to look at our OGTs right now, mine probably looks like that one. It doesn't really change much between people. So that is going to be our, what we'll call like normal, our baseline, our typical, what you would see for a person.
Okay. And I guess on that slide, it looks like there's an overexpression, like the green and under for the blue. Is that normal outcomes? I thought it was always just less so.
So there's going to be variability for sure. That's why we definitely, we do things in at least triplicate, and we run statistics. So the statistics we ran said that if you look at these two, they're not different. Like this is just kind of the variability you get from taking a sample from a big population. Whereas when we looked at this one, our statistics that we ran said that there's no way these populations are the same.
Okay. Great, thank you.
You're welcome.
Hughie has a question.
Maybe another question. Sorry. When you tested glucosamine on the different OGT variants, did you use the CRISPR generated cell line, or just the overexpression and tested the glucosamine?
Overexpression. So we've got our wild type, our variants without the treatment. So just normal media, our kind of baseline. And we also have overexpression of K852M. So this is not something you would see in patients ever. It has zero activity. When we added glucosamine, you can see that one doesn't go up. If you have zero activity to begin with, you add more substrate to try to increase it, it's still going to be zero. But yeah, this is overexpression. We plan to redo these experiments with our knock-ins, but this was just kind of a first test run of this kind of an experiment.
Yeah, thank you.
Yeah, I have a question. So for your experiments, you use male cell lines. How would be it different for female cell lines? I mean, will there be any difference? Is it like completely different process, or we can use all you identified on male lines for females?
So I can tell you one reason that we stick with male cell lines for this is that it means we only have to edit or change one copy of the X chromosome, right? So it means in terms of resources, it's less expensive on us. But it definitely in the future would be worth looking at, variants that we find in female patients. What's going on between, the copy of wild type versus the copy that's been changed? And does that maybe play a role in things like X inactivation. So that's when cells in a female decide which X chromosome is going to be expressed, because you don't need 2 copies, right? Because everyone that's XY only has 1. So it's got to pick essentially which of those 2 copies. And I imagine for female variants that we find in females, there might be some issue there. But in terms of picking it for our experiments, it's just easier. We have, right, for every time that you try to alter a gene, you're only going to get, like some of the ones that we have, 9 or 10% actually get changed, right? But if you take that 9 or 10% and then you multiply it again by 9 or 10% for the other X chromosome, you're then looking at 1% that actually has both copies. And that's really low numbers. And like that 10% is kind of high and good for some, right? So it's more so that it's easier to study.
Yeah,
Okay.
thank you.
Thank you.
I have just maybe one quick update for the statistics. So company GenDx, they do exome sequencing, and they shared the information on how many patients they identified with mutations in OGT, and it was very strange. So they found 11 males and 12 females, which is, I don't know if that's in line with you. I would expect that the amount of males would be higher than females, but that's what they reported to me.
Out of how many people?
So all the people they tested, so basically how it works, like someone needs to refer a patient with some problems for this testing, right? So it's among all the patients that they tested from year 2015, they found 11 males and 12 females with mutation in this gene.
So all 23 of those patients had intellectual disability?
That's, I don't know, that they just tell the numbers for how many mutations did they find in this gene.
And we were surprised with the small number.
Small number, and the fact that females number is higher than males, that's strange. I also have a follow-up questions for them, because for this test they usually test parents and patients, right? So maybe those females, it's just carriers, mom carriers, and not the actual patients, but I'm still waiting for the reply on that.
Sage, please go ahead.
Yes, just to build on that with the X inactivation, and as an affected female, I have a X inactivation ratio of 88 to 12. I hear you guys speaking of the intellectual disability. Of course, Dr. Morava has, now clinically diagnosed me with OGT. However, I have no IQ delay. I do have executive functioning problems and such, but are you guys considering scattered intellectual disability as, a possibility with no IQ delay in females?
Yeah, so we're probably going to miss, we're not going to miss them. We're basic scientists, right? I think a lot of the clinicians will miss them, right? Most likely. Because I understand what he was saying, but it's not, the way they're going to catch those is probably the, I'm going to guess, I don't know your case, but I'm guessing your child got diagnosed first, right? Is that correct?
Well, I was diagnosed when I was much younger, but I was misdiagnosed first, with the mood disorders, versus intellectual disability, or autism or whatnot. Now I'm, they're seeing me as on the spectrum, probably considered level 2 autism, fully based off of my physical disabilities and my support needs for that. But the only delay that I don't have is considered, no IQ delay. They're now seeing, as my sons are getting older, that they are probably likely not going to be IQ delayed either. They just can't accurately measure their IQ right now because they're not able to speak. However, in my family, they have found that is due to aphasia from the other neurological problems and muscular issues and delays that we have.
Oh, that's really interesting. Yeah, it's one of the main ways it gets ruled in is ID, right? And so, because right, it's almost in the name, right? Intellectual disability, and so it'll be interesting to see. That's going to be a diagnosis for the clinicians, not for the researchers. And so I'm not sure exactly how they're going to rule that in or out. That'll be interesting to see.
Well,
Can you,
um,
offline at least, I'd love to know what the mutation is.
we have the we are switching A to G, I believe it is. Hold on, let me pull our papers up. I actually have them right here. Let me see. Yes, we have the yes, we're switching it's the 3 or C dot 3029 A greater than G. We're lysine 1010 to arginine, the arginine arginine, sorry. There is what our variant is. I don't know if
I'm sorry, could you say
that.
is lysine 1010 to what?
To the, is it arginine or
Oh, yeah,
arginine?
it's
Yeah.
arginine. Yeah, lysine
Yeah,
to arginine at 1010.
Mm-hmm.
Oh, that's interesting. Okay.
I mean, I will say for our update on our family, really, we've now gone to University of Michigan Genetics as of it was the end of March now, and they have gotten me to do the full West testing, and my mother now has also agreed to do the whole, exome sequencing and exon activation OGT testing as well, which she'll be doing later this month, or sorry, later in June.
Great.
Yeah, there's definitely a lot we have to learn. And right, the initial 5 that Nithya and Krithika characterized were all in male patients, and we saw that their mothers were unaffected. They had like 95 to 5 skewing, right? And then as we learn more, as more people, as we become aware of more people and more variants that have it, we're definitely learning more. Because, let me go back to it. Ooh. Right? If you'd asked me 5 years ago, I would have said this is an X-linked recessive disorder, right? That you would need 2 copies for a female to be affected. But we know that's not true anymore. And so we definitely have a lot to go. And I think the exon activation definitely is going to be an interesting piece when it comes to especially understanding the changes that are going on between during development, right? That's when a lot of those cells are deciding, this copy or that one, and how that plays out into whether or not, you see ID or developmental delay or these other features.
Okay, thank you very much for that.
Sarah, I remember you unmuted yourself. Did you want to ask anything?
And mine was just more of a general one as to whether there's anything that we can do to help support you guys with your research.
I mean, if, I know one of the things that's been helpful is kind of sharing the history and things like that. I know when Andre and Arena came, I got a new appreciation for kind of the muscle aspect of this, right?
Yeah.
Dinner, Arena was like, feel her arm, and you could feel that there's just not a lot of muscle mass. And you tie that in with the story she told us about early on, not meeting those milestones in terms of moving, kind of gives us new insight, right? So a lot of our models so far are focusing on this intellectual disability, right? So we're looking at nerves, neurons, that kind of, right? But this is a syndrome, so it's going to affect different parts of the body as well, right? And we don't really know what's going on with this here, but definitely more work to be done.
Okay. Yeah, I mean, we went over to Dan's lab and did a presentation. I don't know if that's been shared with you, but we're happy to share that with you if that would help. Or
yeah, of course. You can always send it to Lance. He's kind of our go-to between clinicians and families.
Okay. Thank you.
Yeah, Sage, go ahead.
I just did have one more question for you guys, really about some of the mice studies and stuff, especially considering the eye abnormalities. Have you guys at all looked into hypertensive issues with the brain in the mice at all?
So I don't think we have a mouse model yet. I know, Lance, you probably could speak more to that. I know you're on taking care of that.
Yeah, so we don't have a mouse model yet. We're
Okay.
working on getting some mouse models and actually thinking about a different type of rodent model.
Okay.
But the best person to talk to in terms of mouse phenotypes is going to be the Van Alpen lab, Dan's lab.
Okay. Thank you so much.
Yep.
Sage, do you have any update? In the past, you mentioned that Dr. Eva Morava put you on clinical trial for glucosamine, or was thinking to put you on clinical trial. Do you have any update? Is it started, and what exactly is she measuring on you guys?
So I have not started it myself. Both of the boys have started it. The only real difference that I've noticed in the boys is that there seems to be, in combination, at least for my older son Zachary, when he uses guanfacine for his ADHD, that it seems to regulate his behaviors and anger and such. However, he seems to do just about the same whenever he's not on either the supplement or, the medication together, and we're just well supporting him at home for his ADHD and sensory needs. Theodore, I've noticed at least he has a reduced amount of bruising. He does have some clotting issues that they've noticed in hematology, and Zachary and I also have some mild inconsistent clotting labs as well too, that they're unspecified at this time to any other disorder. However, I have not yet started the glucosamine because I have been waiting to meet with endocrinology to have some clear blood tests. So before, and pretty much finishing all of these tests that Dr. Morava had wanted me to finish before starting the supplementation there. I will say the thing that we found out in the past couple months is there's been, especially with the University of Genetics, is they're more concerned about the muscle issues in me and my sons. We are both, or all of us are very low muscle tone. Theodore, they had suspected when he was younger and when he was born that he may have had a stroke in utero because he has a lot of one-sided weakness. And we all three of us have the one-sided facial, a little slight droopiness, especially with our left eye in all three of us. My mother and my brother also happen to have that affected, which is now why we were looking into that. And I asked that question about the eye abnormalities because it wasn't until recently, I think like last week or so, the my neurologist finally found what he thinks was the cause, but it was silent brain hypertension that has slowly been killing my optic nerves in my brain and causing double vision and any of the other symptoms that I've had there. And they found it only based off of lumbar puncture. They found that my pressure was actually extremely high to the point where they would have assumed if they hadn't seen me in office that I would have been actually a quite obese woman because my pressure was so high. Now the ophthalmologist has seen the same kind of like I said, symptoms, at least in Zachary and Theodore for the eye abnormalities. But, of course, we haven't gone so far as to do that further invasive testing with them. We've just kind of, been watching any sort of progression for their eyes getting worse, which has happened, but very, very slowly on their end, at least.
Yeah, thanks for sharing. I think that this eye thing is very common for all the, like Vicky also has one eye inverted, and I don't know, like it's like after stroke effect, when you have like one eye a little bit closed compared to another one. That's another like thing that I noticed in her.
Yes, yeah, I have had, at least for myself, it is mostly presented as eyelid laziness rather than the actual turning inwards of my eye, as well as actually having, glaucoma type symptoms. I actually think that they're about to change my eye diagnosis to glaucoma with the intracranial hypertension diagnosis now. They were only waiting on that glaucoma diagnosis because they could not find the cause of what was causing my nerve damage for my eye at this time. So there is that. I mean, I wear glasses, of course, so that's helped me a lot. I see that a lot of, of the kids here, they have glasses as well too. So, if you guys have any other questions on that for me though, of course, I'm willing to answer and share any of my experiences or what the kids have gone through.
One other physical thing I can share with you with my daughter, Lauren, it's a little peculiar, but she does not tan. She can lie in the sun for hours and her skin tone does not change. She doesn't burn. She doesn't tan. She's exceptionally low tone, that she can't eat a normal diet, can't chew, but her physically, her skin does not change any color, even with exposure to sun.
That's fascinating. So, has anyone looked to see if she has melanin?
No, that hasn't been checked out.
Oh, that's interesting. Did she see Eva?
Yeah. She did all the blood work with her, so nobody came back with any concerns.
Oh, great. Yeah, I mean, it wouldn't be a concern. It would just, I'm trying to think of an explanation. Right? The failure of melanin to develop, maybe, of the melanocytes, which are, kind of go around with dysmorphic facial features, all of that. And all right, melanin. What makes melanin comes from what are called neural crest cells, which are involved in also making your facial features and part of your brain. And so it's all connected, is what I'm saying, right? Very closely connected. But I'd never heard that before. That's fascinating. That's a really that's an unusual phenotype. Yeah, I've never heard of that. Like, just doesn't tan. Or doesn't burn, right? That's
exactly. Thanks.
Yeah, thank you. Neil, are you planning to do exon activation test for your daughter?
What type test?
It's called exon activation test.
It's like eczema?
No. We can connect online, but probably like it's a female, so Dr. Iva Morava would recommend you to do this. It's called exon activation. It basically shows is it random or non-random. It's when the body shuts down one exon. So the thing is, like all the females who were tested so far, it's non-random and very high skewing. So maybe it was just to do another test on your kid and see how it looks.
Okay, well, yeah, let's circle up and talk about that.
Have any of the other parents noticed inability to tan in response to sunlight?
No.
I would say actually me and my kids have the exact opposite problem where I have a burn right now the other day because I went outside and I went straight from, being pale to actually I tend to get like sun poisoning very, very easily. Though they've always written that off as because I was diagnosed long time ago with the connective tissue disease.
Okay. Yeah, the natural histories are fascinating. That's one of the things as a field we just need to get a much better handle on is what the parents know, we need to know, right? And we need to do it in a very methodical manner, like where everybody's answering the exact same questionnaire. So that's definitely one of the things I think that's kind of coming out of the meeting we had back in November is this need for a really good natural history, which Eva's group could definitely do. Judy Friedovich Kyle could do. Emery, a couple of different groups could do. And so we just got to get everybody on the same page. And so hopefully if the community reaches out to you and if you know other people in the community asking you to fill out a questionnaire, that'll probably take like, maybe an hour, right? Unfortunately, it'll be probably detailed, but it could be really helpful just to see, what's unique to individuals and what's kind of common traits, right, that we see, right? Like the pain thing or the lack of muscle tone, right? That to me, that's all starting to maybe talk about peripheral nervous system issues, which is interesting. So, and just give us a lot more information. So hopefully y'all would be willing to do that if we can kind of get organized and get a natural history study out. So.
I thought, I think it's Marie in Dan's lab was starting to, well, I think she has like an Excel spreadsheet that she was starting to send round. I don't know if that's what you're if we can build on that.
We have a huge Excel spreadsheet with different things, but what we really need is to do it like an MD would do it. I mean, have an MD do it probably, because they're going to capture All right. The thing is, like in different people's diagnosis, it'll say, like, some people will say, the physician will say large head. The other physician says at 6 months of age, 28.2 centimeters. That's a much more useful number, right? And like it's quantitative, right? As opposed to, oh, their ears seem like they're low. Their ears are, this low compared to where their eyes are or whatever, right? Like being very specific about it.
Okay.
And that's part of a natural history, but that requires also the physicians to be on board and like take all of those measurements, right? And so,
Okay.
but no, it's great it's great that the spreadsheet, right? We just need to all be sharing all of our things, all right? We're all
Yeah.
kind of doing it independently, and we just need to get a little bit better organized, I think.
Okay.
And that hopefully Eva's going to take the lead in that. I'm not sure, but that would be my hope, or Andy Edmondson, or Judy Friedrich.
I agree with you because another wrinkle is my daughter, probably because of this mutation, had infantile spasms at 6 months, and it was, we didn't know about OGT, and it was a horrible experience, but it probably was a sideshow. So to this day, we don't know her intellectual disability, how much comes from OGT and how much is lingering effects from the seizure history. I mean, Eva said the way she presented physically, she expected a much higher, intellectual functioning, which isn't there. So that's probably another wrinkle for your research, to make sure you record seizure history, because that could impact intellectual disability alongside the OGT as well.
Yeah, Neil, you're definitely not the first parent I've talked to where they either had seizures or almost an epilepsy type of like continued seizures throughout their whole life. So yeah, I mean, again, right, just getting all that information together and trying to pair it out, right? The more information we can have, the better.
I did want to mention one more thing, since you guys were speaking on the peripheral, nerve issues and everything like that. I have been diagnosed myself with a polyneuropathy. They've kind of written it off as the chronic pain again and connective tissue disease my whole life. But I ended up having issues so bad, especially with my legs and the spastic issues over the years, that I now have had to have a spinal cord implant placed in my legs. In my back, for my legs to be able to work properly and my nerves to be able to work properly at this point. So far, with the only thing that they have been able to tell on the clinical end there, so this is really just more me sharing information, is that I have had repeated episodes of low potassium in combination with calcium and magnesium and iron being low usually. And what I've experienced in those times is my intracranial pressure also increases at the same time, which for me causes seizure-like episodes that end up with me having like dystonia and the double vision issues and one-sided like facial droopiness, and sometimes paresthesias in my face and arms and legs. I will say also, I do tend to have a lot of memory issues and other almost regressive like issues after the fact, because I always struggle a lot more with my ADHD and my executive dysfunctioning after those attacks happen.
So when you say a connective tissue, like, were you initially diagnosed with like Ehlers-Danlos, or?
Yes, I was. Yeah. Mm-hmm.
Yeah. It's when they don't know what to do, they call it Ehlers-Danlos or multiple sclerosis, right?
Yes,
like,
exactly. Yes,
you probably got diagnosed with MS too, right? So
no, so they're actually looking at myasthenia gravis at that one. I have some antibodies for it, but they are not quite enough that they are positive for myasthenia gravis. Yeah. Dr. Morava has actually suspected that there's a possibility that I may have a secondary congenital disorder of glycosylation there. But as of right now, I have not heard back, of course, for the whole exome sequencing, because that is something else that they were looking at the time. Also, a potential hypokalemic paralysis disorder is another thing that they were looking at.
Thanks for sharing all of that. That's great. It's really just useful probably useful for everybody, right? Because everybody has a different journey a little bit. So.
Of course, yeah, no problem.
Sage, and maybe, I don't know if you have time, but maybe you can quickly update how are your boys doing. So Sage has 2 sons, how is it doing? And you mentioned that they don't have IQ issues. Like, are they verbal? Are they potty trained? Do they know colors, numbers?
Yes, I would say they are still a little bit slightly behind, I feel like, but I think that it is more of an attention deficit issue for both my boys because they just, they focus on what they want to focus on, really. Granted, both my boys are very interested in school stuff. So like Zachary, he's coming up on 6. He can read. We're positive of that. He can fully read at this point. But it's really hard to judge just how much he's reading and understanding because the speech delays at this time. He is a Gestalt processor, so we're realizing that he's reading because he'll see something and then that we don't know that he knows, and he's immediately able to repeat it, like like a store sign or something else like that, or other similar things. And Theodore, I think, is actually he's kind of on the same route there. He has just turned 3, but we are highly suspecting that he's reading already at this point. He's actually speaking fairly well now, and he has been on the glucosamine supplement since he was about 18 months old. He's been on it, I think. So Zachary was, a little bit older at this point. He was coming up close to 4 when he started the glucosamine supplement. I like to say all the time that Zachary and Theodore, they're kind of like the tortoise and the hare, where Theodore is very slow and steady on his development without much regression, and then he doesn't speak very much. So it's kind of hard to tell exactly what he's doing until he's just kind of bursting out that skill. And like Theodore knows all of his colors. He can count up to at least 25 at this point. He definitely knows his ABCs and such. So it's still just very hard to judge because their specialists and their therapists at this time have just highly said that they think that the neurological delays with the aphasia, because we definitely have like some jaw issues. They both had tongue ties and lip ties and things like that. So it's just kind of a combination of the other muscular and joint issues around the face and the upper jaw and throat there, they think are causing a little bit more of the lack of speech, along with the attention deficit with that, because they, again, like I said, they just focus on what they want to focus on. So it's kind of hard to, focus them in on, hey, we're doing school stuff and everything. And then we've also had some inconsistency with support in the schools in general, just because we've moved quite a bit, just to become closer to doctors. So.
Thank you. So, Johnathan, Naomi, do you have any other topics to discuss, or parents, any other questions?
I think that's everything Naomi and I have. Is assuming no one else has any questions, I think we're good.
Thanks a lot, to both of you. Just amazing presentation, and it just, every time I feel like I cracked this topic, but every time I'm like, oh, maybe I 20% understand. So way to learn.
Thank you all. Will the slide deck be available on the website or can be sent around?
Yeah, so Andre is in charge of that. He will definitely post the recording of this call, and we will ask for the deck, Johnathan, if we can share it with the group. But Andre will get in touch on the communication after this call.
Okay, great. Thank you, everyone.
Yeah, thank you.
Thank you all.
Thank you. Thanks.
Okay.
Bye.